There are two mechanisms by which enveloped viruses enter host cells. In one of the mechanisms, the virion attaches to host cell receptors by specific proteins on its surface, called spikes. The envelope of the virus fuses with the plasma membrane of the host, and the nucleocapsid is released directly into the cytoplasm. The nucleic acid then separates from the protein coat. In the second mechanism, the enveloped virus adsorbs to the host cell by specific proteins on its surface, and the virion is taken in by endocytosis. In this process, the host cell plasma membrane surrounds the whole virion and forms a vesicle. The envelope of the virion then fuses with the plasma membrane of the vesicle, and the nucleocapsid is released into the host cytoplasm. The capsid protein is then removed, releasing the nucleic acid of the virus. A naked virion also enters by endocytosis. Since the virus has no envelope, it cannot fuse with the plasma membrane. After being engulfed, the viral nucleic acid is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
It does have these organelles, ribosomes, which you know are important for protein production. Here's its singular piece of circular DNA that we see here. It's all folded up. It is one circular ring. And here's an extra small plasmid, a smaller piece of DNA, which is holding non-essential genes. We can also see the flagella, which help in the motility of the bacteria. Let's quickly run through some more of the structure of the bacteria. Again, they're prokaryotic, lacking a nucleus, lacking large membrane-bound organelles. They do have a cell wall made of a carbohydrate called peptidoglycan. That's significant because we know that all cell walls are made of carbohydrates. In plant cells, they're made of cellulose, and in fungus cells, they're made of chitin. Just a different type of carbohydrate here. When we look at bacteria, there's one way to kind of easily differentiate between different broad groups of bacteria, and that's whether they are gram stain positive or gram stain negative. The gram stain is a chemical stain that you can apply to a sample of bacteria, and the ones that are gram stain positive will show up dark purple, as we see here and here, and the ones that are gram stain negative will show up in this pink color. And this helps us differentiate between known types of bacteria. And the reason why they stain differently has to do with the structural difference in their cell membrane and cell wall. The bacteria that are gram negative have a cell membrane, a thin cell wall, and then another membrane on the outside, whereas the gram stain positive bacteria have a cell membrane and a thicker cell wall that reacts with this chemical stain differently. Another way we can differentiate between bacteria is their basic shape, and there are three basic bacterial shapes: spirili, bacilli, and cocci. Spiral, rod-shaped, and spherical. Oftentimes, these names, spirili, bacilli, and cocci, are built into the scientific name for that bacteria. You'll definitely need to be able to match these names, these terms with these shapes, and by site and by words here. And we talked briefly a minute ago that many bacteria are motile. They have a long whip-like tail called a flagella. Some do. Sometimes they have a simple one singular flagella, sometimes multiple. They can have them on both ends to go one direction or the other without turning around. And sometimes they have many coming off on all different sides. The point here is that the bacteria
Nuclear membrane is absent in prokaryotic cells. There is no well-defined nucleus in prokaryotic cells. Only nuclear material, nucleoid, is present. Well-defined cell organelles like mitochondria, endoplasmic reticulum, etc. are absent in prokaryotic cells. Prokaryotic cells are found in bacteria and blue-green algae. The eukaryotic cells have a well-defined nucleus. The nuclear membrane and nucleolus are present in a eukaryotic cell. Well-defined cell organelles like mitochondria, endoplasmic reticulum, etc. are present. Eukaryotic cells are found in most plants and all animals.
Viruses are tiny parasites that reproduce inside the cells of a host. The host body provides the materials that the virus needs to survive and replicate. In humans, viral infections cause illnesses ranging from the common cold and flu to hepatitis and AIDS. Infection can be caused when virus particles, called virions, become active inside a host. The virions, which consist of nucleic acid surrounded by a coat of protein, transfer their nucleic acid to adjacent cells. This creates new proteins and new virions, which can start another cycle of infection and spread throughout the host. But viral infections can often be prevented by vaccination. A vaccine consists of a dead virus which causes the body's immune system to form appropriate antibodies. Antibodies are proteins found in the blood that attach themselves to any foreign substance and neutralize it. Medical researchers are constantly seeking new vaccines to fight disease-causing viruses.